Welcome to MOOC course on Introduction to Proteogenomics. In the last lectures, Dr. Kelly Regals have given you very detailed overview of genomics, transcriptomics and epigenomics. Continuing in the same theme, let us talk about SNPs. SNPs are most common type of gene polymorphism and they are located in the promoter regions of genes, thus the bringing the changes in gene expression. Today's lecture will be given by Dr. Bing Zhang, who is a professor of molecular and human genetics at Baylor's College of Medicine in USA. Dr. Zhang will introduce you to the concepts of DNA polymorphism and how they bring about the variability in a given population. The lecture also aims to provide an understanding on how genotype could influence the trait of a phenotype. So, let us welcome Dr. Bing Zhang for today's lecture.
So, I will start with a simple definition of genotype. We know that genotype refers to the unique genetic makeup of individual organisms and it's encoded in the DNA sequence, we know that, right? So, the difference in the DNA sequence between individuals is called DNA polymorphism. And if we look at this example and let's say this sequence, you can probably download from the let's say the human genome sequence and it's just a short fragment, right? We can see some individuals like this guy has exactly the same sequence as a reference, but this guy for example has a different nucleotide at this position. And then if we look at this position, and there are two possible alleles, one is the G, the other is the T, right? So, so this is a biallelic locus, meaning there are two different types of alleles. And then the major allele is the G because it occupies 80% of this very small population, right? And the minor allele is the T, which is only 20%, 1 out of 5. And there is another biallelic locus here, where we found two individuals have the G, where all the others have A. And then this is also a biallelic position, and with the major allele 60% and the minor allele 40%. So this is a very simple type of DNA polymorphism, and because the difference only occurs in a single nucleotide, it's called single nucleotide polymorphism or SNP. And this is a very common type of DNA polymorphism in the genome, and it's very frequent. For example, in the human genome, probably there are around 10 million SNPs in our genome. And there are other types of DNA polymorphism like deletions, insertions, or even copy number alterations for large fragments. So, and the phenotype is obviously the observable traits or characteristics of individuals. And depending on the nature of the phenotype, and it could be binary phenotypes, meaning there are only two possible selections, like any disease. You are either diabetes or non-diabetes. Or it could be continuous in nature, like the quantitative traits, like our height or weight. And these are continuous or quantitative traits. And the phenotype, especially if we're interested in this, for example, we're interested in the disease phenotypes, right? And it's usually the interaction between the genotype and the environment and their interaction. And the genotype plays a very important role in determining one's phenotype. So that's why a major goal in biomedical research is to understand how the genotype determines the phenotype. And in order to do this, in a very simple but powerful way is through the association analysis. So in the association analysis, what we want to do is, if we are interested in a phenotype, we want to test whether there is an association between the genotype and the phenotype. And depending on whether the phenotype is a binary phenotype or a quantitative phenotype, we need different types of statistical tests in order to establish that relationship. So let's first look at the binary traits. And this is usually referred to as case control studies. And for example, disease is case and non-disease is control, right? And if you have a population or a sample size of n, and then some of them are disease samples or case samples, some of them are control samples, right? And then some of them have this genotype and this genotype and this genotype. So basically, you can look at your individuals and put the numbers into these 2x3 tables. And so basically, and these are the cases with this small a, this genotype, right? You can fill in this table very easily after you, if you know all the genotype and the phenotype for that, all the individuals. And then we can use a very simple chi-square test in order to test the association between the binary phenotype and the genotype. And this is a formula showing how chi-square test can be done, right? But sometimes, when for example, the major allele might have a dominant effect, that means whether you have this heterozygous locus or this homozygous locus, you are going to have the same phenotype. So, and then in that case, we can combine these two into the same column in the table, and the 2x3 table can become a 2x2 table, right? By doing this, we actually gain power because we reduced the degree of freedom from 2 to 1, and then if the phenotype is actually, the major allele has a dominant effect, we actually gain power by doing that. And on the other side, if under a recessive model, and we would expect these two, and then only the A, this one will have the same effect, but these two will have the same effect. And then again, we can combine those two columns into one, and then we can do the chi-square test. But sometimes maybe there is an additive effect. Let's say if whether you are in this genotype or this genotype. So let's say the count, the number of the minor alleles is kind of linearly associated with the proportion of the case or the disease proportion in the population like this. And then if we simply do the chi-square test, we won't be able to capture that relationship, right? So what can we do? So people have come up with this, it's called a Cochrane-Armitage trend test. So with this test, by playing with the weights, the W's in the formula, so you will be able to, for example, if you set the W as 0, 1, 2, you will be able to test this additive effect with the formula. And the interesting thing about this test is that you can change the weight setting to 0, 1, 1 or 0, 0, 1, and then you will be able to also test the dominant model and the recessive model. So usually, we don't really know which model SNP or phenotype is determined, right? And one way you can do is to test all the possible models, and then you get the most significant one and use that for your report. And another way to deal with this additive effect is you can consider converting the genotype count table into an allele count table. So basically, if each person will contribute 2 alleles to your count table, so your sample size actually increase from n to 2 times of n, right? And then you count if one have heterozygous, then allele locus, and then it contributes to both of the slots in this table, right? So that way you can still do the chi-square test, and the degree of freedom is still 1 for this. But this also assumes the additive effect. So those are for the binary traits. But for the continuous or the quantitative traits, and for example, the blood pressure or cholesterol levels, this type of things, what we can do is to fit a linear regression model against the data. So basically, the covariates will be the number of minor alleles in individuals, and then you correlate with that the continuous measurement of the phenotype, for example, blood pressure, and then you can test the goodness of fit of that linear regression. And if you find a locus is actually associated with a quantitative trait, we call that locus a quantitative trait locus, or QTL, and probably you have heard about QTL many times. And so far we have been talking about just you are interested in one SNP and one phenotype, and then you try to establish the relationship. But oftentimes, I mean, you start with a disease of interest or phenotype of interest, and then you don't really know which SNP or which position of the DNA sequence is associated with that phenotype, right? In order to find out, you have to scan the whole genome, the whole DNA sequence, and all the SNPs in order to find which one is actually associated with that phenotype, right? In order to do that, we have to do the genome-wide association study, which is a GWAS. I think, again, probably you have heard about this term already. And in order to do the genome-wide association study, we first need a very kind of low-cost assay or platform in order to do the whole genome genotyping. And this can be done by array-based platforms. And nowadays, the arrays can go up to 1 million SNPs. So basically, you can scan 1 million SNPs at a time. And you can also go with the next generation sequencing-based approach, because the array-based approach, we are talking about the 10 million SNPs in the human genome, but the array can only cover 1 million, right? But if you do a next generation sequencing with a very good depth, and then this is truly unbiased assay, but it's still more expensive than the array-based technology. And then you also need a large population with both case and controls, or with different variation for quantitative trait. And then, of course, we need the statistical analysis in order to analyze the data. But the basic statistics is simple, is the one we just talked about, either the chi-square table test, the trend test, or the linear regression test for the quantitative trait. And one thing we want to remember is, because now you are testing a million SNP, for example, against one phenotype, and then you are doing a lot of tests. So this is the multiple test problem. I think Dr. Mani has talked about that yesterday. So we need to be careful to adjust for the multiple tests, because I mean, you did so many tests, and by random chance you are going to get some positive hit, right? We want to make sure that what we get is not because of that reason. So, and when you get the result from the GWAS analysis, it can be visualized in this Manhattan plot. So in this plot, so on the x-axis, these are the genome locations of the locus. And each chromosome is indicated by a different color. So here is chromosome 1, and here is chromosome 22. Usually we don't include the sex chromosomes here. And on the y-axis, this is p-values in the minus log scale, because if you take minus log, and then the smaller p-value will have a bigger value, right? And then it will appear at the top of the plot. And then immediately, this is a very effective way for visualization, and immediately you can see at this chromosome 3 and this chromosome and this chromosome, the 3 chromosomes at these positions, there are kind of the SNPs are associated with the phenotype you are interested in. So that's very effective way to visualize and understand your data. And well, so the GWAS catalog is a resource that captures all the the SNPs associated with different kinds of diseases or phenotypes, categorized into, I think, 17 categories. and then you have all the associations in that resource. And then they actually used the p-value of 5 multiplied by 10 to the minus 8 in order to determine the significance. So can anyone tell me why they pick this cutoff rather than just 0.01 or other values? The 0.05 is 5 times 10 to the minus 8. Why that is used as a cutoff? So if you are going to select the significance from a test, what p-value are you going to use? 0.05, right? But why they use such a stringent? Because this is 0.05 divided by 1 billion, right?
To reduce the number so we can get specific traits, not so many findings.
Because there are too many findings, you want to get the most significant ones?
Correct.
Any other answers?
So here when we are going for a whole genome sequencing, then maybe we are doing multiple reads, because of which we want to make sure that very less and very, like, there are no variation in between. So if variation in there, we need to remove it. That's why we are going for a more stringent criteria.
Okay, it's because of the variation, you think?
Yes, because multiple reads are being taken here.
Yeah,
There
yeah,
can be some
exactly. So it's a multiple testing we're talking about. And the reason they pick this value is because most of this data were generated on the SNP arrays, which measure a million SNPs. That means you have a million times of tests. So they just did a very simple bufferonic correction, as Dr. Mani mentioned yesterday. So basically, you have to use a cutoff. So basically 0.05, as you typically do if you only have one SNP, you have to divide that by 1 million. So that's why you get this number. Yeah. So I think so far, we talk about very basic genetics and the GWAS studies or association studies. I think most of you probably have already been familiar with those. So the next part, I'm going to talk about the molecular traits as quantitative traits. That is probably more relevant to this audience. So we know, although we are interested in the association between DNA and the phenotype, but we also know that there is a lot of other things going on between the genotype and the phenotype. That means the DNA has to be transcribed into RNA, and the RNA has to be translated into proteins. And there are a lot of regulations going on, right? When you have DNA, you not end up with the same protein, you not necessarily end up with the same phenotype. So in order to understand this whole process, we can consider the gene expression measurements as also as quantitative traits. If you think about if you do an RNA-seq experiment, for example, and for 100 patients, and then you are going to get gene expression measurement, for example, TP53 abundance 100 times. And then that is actually a quantitative, you can think that as a quantitative trait, right? And then if you also have the genotype data from the same cohort, and then you can actually correlate those. And if you do that, this is called the mRNA abundance as quantitative trait, or its expression QTL or eQTL. So basically you are trying to establish the relationship between a genotype, but against the gene expression, not the final disease phenotype. I think yesterday somebody asked about the ChIP-seq, right? You can also do ChIP-seq in order to get the actively translated region of the RNA, and then so this indicates the ribosome occupancy. And then if you associate those measurements with the genotype, and then if you find the QTL, it's called the RQTL or ribosome occupancy QTL. And similarly, we can do protein abundance measurements, and then we can associate the genotype with the protein expression, and then if we get an association, it's called pQTL. So in these studies, usually when you do a disease study, you have one phenotype of interest, and then you scan for many SNPs. But this time you have 10, maybe 10,000 genes with all the measurements, and then so basically it further increase tests you can do. It's very powerful because you can test so many hypotheses at the same time, but just be careful about your multiple test adjustment. But the beauty is that you can understand the regulatory mechanism for all the SNPs and all the gene or protein expression. That's pretty cool. But the basic is very simple. I mean, because you treat each gene expression or protein expression as a phenotype, quantitative traits, and then you can do the same linear regression and to find the relationship. So I'll show some, before we go to some examples, I want to mention there is a difference between the typical disease phenotype and the gene expression or protein expression or the ribosome occupancy, this type of phenotype. Because both of them, like the genes and the SNPs, they have the location relationship on the genome. And then sometimes a SNP can be very close to the gene. And a lot of times you can find SNPs in the promoter region because there are a lot of regulatory sequences in that region. And then you can find the relationship between genotype in that region and the gene expression right next to that SNP. And these are called the cis-EQT errors. Or similarly, I mean, if it's protein expression, it's called cis-PQT errors. And as you can see in this example, if you have a SNP here, it may, especially in the promoter region, and it may affect the expression of this gene. That's very understandable, right? And it has been shown that the cis-EQT error is very important because the disease-associated SNPs are enriched in these cis-EQT errors. And let's assuming this gene actually encodes a protein which is a transcription factor. And then it could have additional effect on the other proteins, not in the same region, but maybe farther away from the SNP, or even on another chromosome. You can still find that relationship. And in this case, it's called the trans-EQT errors. That means the gene expression or protein expression you are interested in is further away from the SNP you identified. Usually this effect is, because it's a relatively indirect effect, it's relatively weaker, so usually you need larger sample size to identify those. That's why there are fewer like trans-QT errors that have been reported so far. But if you do find some of them, it's very nice because it will show you maybe how one SNP may alter a pathway or network, because the multiple genes might be controlled by the same SNP that can tell you how the network, regulatory network works.
In today's lecture, you were introduced to the association analysis, which is used to understand the relationship between genotype and phenotype. Various statistical tests could be used to understand the relationship depending on whether the traits are quantitative or binary. Manhattan plots could be used to understand the results when GWAS or genome-wide association studies are performed. Depending on the position of single nucleotide polymorphism SNPs and the gene QTLs, it could be either cis-QTL or trans-QTL. Those could be analyzed using these tools. In next lecture, you will be given concepts of the power of integrative GWAS and eQTL analysis using various examples from literature. Thank you.